resources like seeds, fertilizers, and pesticides, agricultural inputs, equipment used for agricultural production, farm machinery, transforming and selling agricultural products, processing and marketing, practices to protect natural resources in agriculture, environmental conservation, study of crop cultivation, improvement, and management, crop science, development of plants from seed to maturity, plant growth, study of heredity in croplands, crop genetics, study of plant functions and processes, crop physiology, farming that meets current needs without harming future, sustainable agriculture, developing new crop varieties and improving yields, research and innovation, knowledge dissemination to enhance farmer productivity, extension services, reliable access to sufficient food for all, food security, raising animals for food and other products, livestock management, commercial activities related to agriculture, agribusiness, impact of climate change on agricultural practices, climate change challenges, land suitable for growing crops, arable land, projected increase to 9.1 billion by 2050, Philippine population growth, agriculture's share of the national economy, gross domestic product contribution, annual food supply growth of around 2%, food supply increase, improvements in irrigation and crop protection, technological advances, Philippine law to modernize agriculture sector, Agriculture and Fisheries Modernization Act, use of scientific principles to enhance yields, crop production as science, practical skills applied in successful cultivation, crop production as art, growing crops for market profit, crop production as business, economically cultivated plants for human use, crops, investigation to improve farming practices, agricultural research, maintaining diverse species in agriculture, biodiversity preservation, methods to protect soil from degradation, soil erosion prevention, systematically accumulated and tested knowledge, science, organism belonging to the kingdom Plantae, typically possessing photosynthetic ability, plant, cultivated plants grown for profit, often in groups or populations, crop, research on plant breeding and genetics, IPB, research on crop protection and pest management, NCPC, conservation of plant genetic resources, NPGRL, research on post-harvest handling of horticultural crops, PHTRC, research in molecular biology and biotechnology, biotech, focuses on research related to fiber crops, FIDA, research related to tobacco production, MTA, research and development for rice production, PhilRice, research focused on coconut production, PCA, research and regulation in the sugar industry, SRA, research and training for root crops, PRCRTC, root crop research and training in northern Philippines, NPRCRTC, research related to abaca, a fiber crop, NARC, dedicated to improving rice production and addressing issues related to rice security and sustainability. IRRI specializes in research for drillland agriculture, including crops like wheat, barley, legumes, and forages. ICARTA, renowned for its contributions to maize and wheat research. CIMMYT focuses on tropical crops like cassava, yams, bananas, and maize. IITA specializes in potato research, aiming to enhance potato production. CIP conducts research on agroforestry systems that integrate trees with crops and livestock. ICRAF focuses on improving crop production in semi-arid regions. ICRISAT specializes in vegetable research, developing improved varieties of vegetables. AVRDC conducts research on various tropical crops to address food security and environmental challenges. CIT focuses on the conservation and utilization of plant genetic resources. Biodiversity International attempts to group plants based on their inherent biological similarities and relationships. Natural system based on the evolutionary relationships between plants, grouping crops according to their genetic and evolutionary histories. Phylogenetic system serves as a systematic way to organize the diverse array of agricultural plants. Importance of crop classification provides a standardized way to name and categorize plants. Standardization, the artificial system of classification groups crops based on practical and convenient features or characteristics. Artificial system. Proper classification helps in identifying plants accurately, regardless of local names or dialects. Identification. Crop classification aids researchers and breeders in studying and improving plants. Research and breeding. Vines are plants that lack self-supporting stems and instead climb or sprawl using other structures for support. Vines. Classification assists farmers in managing crops effectively by grouping similar plants with shared characteristics. Crop management. Shrubs are plants with multiple main branches and no central trunk. Shrubs. It provides a structured framework for teaching and learning about different crops, their characteristics, and uses. Education. Trees have a single central stem from which branches and leaves extend. Trees. The time it takes for a plant to complete its growth, reproduction, and death. Life cycle. Annuals complete their life cycle within a year or less. Annuals. Biennials require 2 years or 2 growing seasons to complete their life cycle. Biennials. Herbs are plants with soft, succulent stems that lack woody tissues. Herbs. Perennials do not die after flowering but live for multiple years. Perennials. Saprophytic plants grow in environments rich in decaying organic matter. Saprophytes. Plants that reproduce sexually produce seeds through the processes of meiosis and fertilization. Sexual reproduction. Plants that reproduce asexually do not involve meiosis or the fusion of gametes. Asexual reproduction. These crops are primarily grown for their edible grains, which are rich in carbohydrates. Cereal/grain crops. Legume crops are cultivated for their edible pods and seeds. Legumes. Parasitic plants establish a connection with a host plant to obtain water, nutrients, and sometimes even carbohydrates. Parasites. Root crops are cultivated for their enlarged roots or tuberous roots, which are rich in carbohydrates. Root crops. Epiphytic plants grow on the surface of other plants for physical support but do not derive nutrients from the host. Epiphytes. Crops in this category are cultivated for extracting sap or latex from their trunks or stems. Latex and resins. These crops are grown for the fibers present in their stems, leaves, or seeds. Fiber crops. Oil crops are cultivated for their oil-rich seeds or fruits. Oil crops. Sugar crops are grown primarily for their high sugar content. Sugar crops. These crops have properties that make them useful for medicinal purposes or as sources of natural pesticides. Medicinal and poison crops. Vegetables are edible parts of plants that are consumed as part of a meal. Vegetables. Fruits are botanical fruits that are typically sweet and eaten raw, cooked, or processed. Fruits. Crops providing nutrition for grazing animals. Pasture/forage crops. Plants grown for aesthetic and decorative purposes. Ornamentals. Crops cultivated for non-alcoholic drinks. Beverage crops. Crops adding flavor and scent to products. Spices, condiments, essences. Surround cell, regulates substance passage. Cell membrane. Network of membranes controlling ion and molecule flow. Endoplasmic reticulum. Rigid structure providing support and protection. Cell wall. Modifies, sorts, and packages proteins and lipids. Golgi apparatus. Gel-like substance holding organelles in place. Cytoplasm. Site of photosynthesis in plant cells. Chloroplasts. Powerhouses generating ATP through respiration. Mitochondria. Houses genetic material and controls cellular activities. Nucleus. Structure assembling ribosomes within the nucleus. Nucleolus. Maintains turgor pressure and stores nutrients. Vacuole. Organelles for photosynthesis and pigment storage. Plastids. Cellular machinery for protein synthesis. Ribosomes. Layer cementing adjacent plant cells together. Middle lamella. Thin, flexible layer allowing cell growth. Primary wall. Rigid layer providing additional cell strength. Secondary wall. Flowing movement distributing materials within the cell. Cytoplasmic streaming. Selectively permeable membrane controlling substance movement. Plasmalemma/plasma membrane. Single-membraned organelle serving various functions. Microbodies. Studied with ribosomes for protein synthesis. Rough ER. Lax ribosomes, involved in lipid synthesis. Smooth ER. Network of proteins providing cell structure. Cytoskeleton. Pigment storage, attracting pollinators. Chromoplasts. Non-pigmented plastids for storage. Leucoplasts. Leucoplasts storing starch. Amyloplasts. Leucoplasts storing proteins. Proteinplasts. Leucoplasts storing fats and oils. Elaioplasts. Cylindrical structures about 24 nanometers in diameter, made of tubulin. Microtubules. Actin chains in a helical arrangement aiding cytoplasmic streaming. Microfilaments. Membrane-bound storage centers for water and substances. Vacuoles. Regions where active cell division occurs in plants. Meristematic tissue. Located at tips of stems and roots, producing primary meristems. Apical meristem. Found in stems and roots, contributing to secondary growth. Lateral meristem. Produces secondary vascular tissues, xylem and phloem. Vascular cambium. Core cambium forming protective outer bark layer. Phyllogen. Present in internodes and bases of young leaves. Intercalary meristem. Mature tissues performing specific functions in plants. Permanent tissue. Plant tissue providing structural support with thickened cell walls. Parenchyma. Outermost layer containing stomata and trichomes. Epidermis. Hard, tough cells providing mechanical support in plants. Sclerenchyma. Long, thin cells with thick walls, contributing to tissue strength. Fibers. Thick-walled cells found in hard structures like seed coats. Scleretes. Protective outer tissue, impermeable to water and gases. Cork. Conducts water and minerals upward in the plant. Xylem. Distributes dissolved food materials between production and utilization sites. Phloem. Responsible for support, storage, and food manufacture. Ground tissue. Protective layer shielding apical meristem during root growth. Root cap. Forms protective outer covering and regulates gas exchange. Dermal tissue. Process initiated under favorable conditions for seed development. Seed germination. Occurs after pollination and fertilization, containing seeds. Fruit development. Tissue responsible for transport in plants. Vascular tissue. Region of continuous cell division for growth. Meristematic region. Area where root cells rapidly elongate. Region of elongation. Transition area for root cell specialization. Region of maturation. Finger-like projections increasing surface area. Root hairs. Single, deep root with lateral branches. Taproot system. Main root growing downward from radicle. Primary root. Roots growing from non-radical plant parts. Adventitious root system. Cluster of similar-sized roots from primary root. Fibrous root system. Roots developing from leaves or petioles. Foliar root system. Supports plant, conducts substances, hosts leaves. Stem. Underground stem storing nutrients, surrounded by scales. Bulb. Flowering stem of grasses supporting inflorescence. Comb. Shoot produced from base of the stem. Tiller. Short branch developing into a new plant. Offset. Horizontal underground stem producing roots and shoots. Rhizome. Above ground stem producing new plantlets at tips. Runner. Shoot arising below ground from old stems. Sucker. Coiling branch aiding in climbing support. Tendril. Swollen underground stem storing nutrients. Tuber. Solid, fleshy stem base serving as storage. Corm. Outer protective layer of the stem. Bark. Inner region of stem, mainly xylem tissue. Wood. Central soft region of the stem. Pith. Part of leaf attached to the stem. Leaf base. Rapid increase in root length during growth. Elongation. Roots extending from primary root for support. Lateral branches. Living substance within plant cells. Protoplasm. Creeping stems include runners, stolons, offsets, or suckers. They crawl along the ground, forming new plants where nodes make contact with the soil. Creeping stem. The petiole is the stalk that connects the leaf blade to the stem. Petiole. Climbing stems have specialized structures, like tendrils, that attach to objects or surfaces. Vines are an excellent example of climbing stems. Climbing stem. The leaf blade is the expanded, green portion of the leaf where photosynthesis primarily occurs. Leaf blade. This type of venation forms a network of veins throughout the leaf, often seen in dicotyledonous plants. Reticulate venation. Parallel venation consists of veins running parallel to each other, commonly observed in monocotyledonous plants. Parallel venation. The flower is the reproductive structure of flowering plants, playing a crucial role in sexual reproduction. Flower. Leaves contain chlorophyll and other pigments that capture sunlight to convert carbon dioxide and water into glucose and oxygen through photosynthesis. Food manufacture. The calyx is the lowermost whorl of modified leaves that protect the flower bud before it opens. Calyx. Leaves exchange carbon dioxide for oxygen during photosynthesis and respiration. Gas exchange. The corolla is the whorl of petals located above the sepals. Petals often serve to attract pollinators. Corolla. The gynecium or pistil is the female reproductive part of the flower, consisting of one or more carpels. Gynecium. Leaves release water vapor through small pores called stomata. This process, called transpiration, helps regulate water loss and maintain a water-conducting column in the plant. Evaporation of water. The gynophore is the step of a pistil or carpel. Gynophore. The pedicel is the stalk that supports the flower. Pedicel. The developing plant inside the seed, including cotyledons, a root cap, a food source, and a plumule. Embryo. The perianth is the collective term for the combined calyx and corolla. Perianth. The scar on the seed coat where it was attached to the plant stalk during development. Hilum. Petals are the individual units of the corolla. Petal. A small pore in the seed that allows water absorption. Micropyle. Sepals are the individual units of the calyx. Sepal. The stem part of the sprouting plant above the root and below the cotyledon. Root. Stamens are the male sporophylls within the flower, consisting of an anther and a filament. Stamen. The outer protective layer covering the seed. Seed coat. The embryonic leaf within the seed. Seed leaf. A complete flower possesses all four major parts: sepals, petals, pistil, and stamen. Complete flower. The shoot of an embryo, which will give rise to the aerial portion of the plant. Plumule. An incomplete flower lacks one or more of the major parts: sepals, petals, pistil, or stamen. Incomplete flower. Orthodox seeds are well suited for drying and long-term storage. They naturally dry out to a low moisture content while still attached to the mother plant. Orthodox seeds. They lose viability more rapidly at low temperatures compared to warm temperatures. Temperature sensitivity. These seeds can be dried to low moisture levels without damage. Drying tolerance. Recalcitrant seeds do not naturally dry out on the mother plant and are shed in a moist condition. Recalcitrant seeds. They are killed if their moisture content is reduced below a critical level. Moisture sensitivity. These are collections of simple fruits that develop from the apicarpous pistil of a flower. Aggregate fruits. Develop from a number of flowers in an inflorescence. Multiple or composite fruits. Intermediate seeds fall between orthodox and recalcitrant seeds in terms of their drying and storage capabilities. Intermediate seeds. Fleshy fruit with papery carpels inside. Pome. Juicy fruits divided into several subtypes. Fleshy fruits. Fruits categorized by opening mode and dispersal. Dry fruits. Fruit with entirely fleshy ovary and many seeds. Berry. Do not split open at maturity. Indehiscent fruits. Single-seeded fruit with loose pericarp. Bacine. Leathery rind fruit with juice-filled segments. Hesperidium. Seed tightly fused to pericarp. Caryopsis. Hard rind fruit with fleshy inner matrix. Pepo. Many-seeded fruit from multiple united carpels. Capsule. Single-seeded fruit with membranous wing. Samara. Variant capsule found in mustard plants. Salique. Hard, one-seeded fruit. Nut. Capsule with a lid that falls off. Pyxis. Loose ovary wall around the seed. Utricle. Small version of a nut. Nutlet. Split open upon maturation to release seeds. Dehiscent fruits. Single carpel fruit with 2 longitudinal sutures. Legume. Single carpel fruit that splits along one suture. Follicle. Fruit develops without fertilization occurring. Parthenocarpy. Requires pollination for fruit development. Stimulative parthenocarpy. No pollination needed for fruit development. Vegetative parthenocarpy. Organelle where photosynthesis occurs. Chloroplast. Pigment capturing light energy for photosynthesis. Chlorophyll. Converts light energy into chemical energy. Photosynthesis. Light energy stored as glucose. Energy conversion. Oxygen released as a byproduct of photosynthesis. Oxygen production. Primary means for plants to produce food. Food production. Remove CO2 from the atmosphere. Carbon fixation. Phase where light energy is captured. Light-dependent phase. Biological catalysts regulating photosynthesis reactions. Enzymes. Influence photosynthetic capacity via chlorophyll types. Genetic factors. Rapid phase requiring light for photosynthesis. Light-dependent phase. Light-independent phase fixing CO2 into carbohydrates. Calvin-Benson cycle. Young leaves have higher photosynthetic efficiency. Leaf age. Plant parts demanding carbohydrates, influencing photosynthesis. Sinks. Essential for nutrient transport and turgor pressure. Water content. Affects gas exchange through stomatal openings. Wind velocity. Larger biomass correlates with higher photosynthetic rates. Plant size. Blue and red light most effective for photosynthesis. Light quality. Higher intensity increases photosynthesis until saturation. Light intensity. Longer exposure increases cumulative photosynthesis. Light duration. First respiration stage producing pyruvic acid and ATP. Glycolysis. Processes pyruvic acid in mitochondrial matrix. Krebs cycle. Produces ATP via proton gradient in mitochondria. Electron transport system. Essential for oxidative metabolism and ATP production. Oxygen and respiration. Occurs without oxygen, less efficient energy production. Anaerobic respiration. High CO2 can inhibit respiration by affecting gas exchange. CO2 levels. Active tissues respire more than dormant ones. Physiological status. Higher moisture increases respiration rates in tissues. Moisture content. Respiration rates double with every 10 degrees Celsius rise. Temperature effect. Water loss from plants, crucial for hydration. Transpiration. Water loss through the leaf's waxy cuticle. Cuticular transpiration. Transpiration aids in moving nutrients from roots. Nutrient transport. Transpiration cools plants by evaporative heat loss. Cooling mechanism. Movement of water through plant tissues via cohesion. Capillary action. Attraction between water molecules aiding movement. Cohesion. Attraction between water molecules and plant surfaces. Adhesion. Pores in bark for gas exchange in woody stems. Lenticels. Water vapor loss through lenticels. Lenticular transpiration. Pressure created by transpiration, drawing water up. Negative pressure. Main type of transpiration, 90% water loss. Stomatal transpiration. Tiny openings regulating gas and water exchange. Stomata. Movement of organic compound through phloem. Translocation. Tissue producing excess assimilates for transport. Source. Tissue consuming assimilates for growth and energy. Sink. High humidity reduces transpiration rates. Relative humidity. Higher temperatures increase transpiration rates. Temperature. High levels can close stomata, reducing transpiration. CO2 concentration. Utilization of produced food by plants. Assimilation. Difference in solute concentration driving translocation. Concentration gradient. Ability of sink tissues to accumulate assimilates. Sink strength. Distance affects assimilate movement efficiency. Proximity to source. Links between source and sink tissues influencing transport. Vascular connections. Internal cycles not influenced by environment. Endogenous rhythm. Yearly recurring events align with seasons. Annual rhythm. Chemicals disrupting metabolic processes affecting translocation. Metabolic inhibitors. Lack of minerals affecting sucrose movement. Mineral deficiencies. Regulate plant processes, including translocation. Hormones. Size and distribution impact transpiration rates. Morphology of leaf stomata. Events that reoccur with each new moon. Lunar rhythm. Events that reoccur approximately every 24 hours, often associated with daily light-dark cycles. Circadian rhythm. Growth is a continuous, irreversible change over time, accompanied by an increase in size, weight, or mass of an organism. Growth. Differentiation refers to the process by which cells acquire specialized structures and functions. Differentiation. Folding of leaves in response to light, often with a rhythmic pattern due to interactions with the biological clock. Nyctinasty. Folding and rolling of leaves in response to water. Hydranasty. Response to touch or mechanical stress. Thigmonasty. Response to shaking without direct contact with the organism. Seismonasty. These movements are directional and are determined by the direction of the environmental stimulus. Tropic movements. Growth towards or away from a light source. Phototropism. Growth in response to gravity, leading to upward or downward bending of plant organs. Gravitropism. Movement of plant organs, such as leaves, to maintain an optimal angle with the sun's position throughout the day. Solar tracking. This type of adaptation involves the development of specialized organs that perform non-typical functions. Morphological adaptation. Nastic movements are non-directional movements triggered by environmental stimuli but not necessarily oriented towards or away from the stimulus. Nastic movements. Upward bending of leaves. Hypernasty. Downward bending of leaves. Epinasty. Physiological adaptations are mechanisms that help plants cope with challenging conditions. Physiological adaptation. Biochemical changes are seen in response to various stressors. Biochemical adaptation. Auxins promote cell elongation, influencing growth and curvature of plant organs. Auxin. Gibberellins are compounds that promote stem elongation, stimulate germination, and regulate plant development. Gibberellic acid. Ethylene is a unique phytohormone as it occurs in a gaseous state. Ethylene. Growth is determined by the scarcest resource, whether it's climate, soil nutrients, biological interactions, or genetic factors. Liebig's law of minimum. The rate of the process is limited by the slowest factor among multiple factors that influence it. Blackman's theory of optima and limiting factors. ABA is synthesized from mevalonic acid, primarily in mature leaves, especially in response to water stress. Abscisic acid. When all but one limiting factor is adequate, plant growth responds proportionally to the limiting factor. Mitscherlich law of diminishing return. ABA plays a crucial role in regulating various physiological processes, particularly those related to water stress and dormancy. Effects of ABA. Seeds placed in moist medium with temperature variations to soften the seed coat. Stratification. Cold treatment prior to germination. Vernalization. Aseptically germinating the embryo removed from the seed. Embryo culture. Hypocotyl elongates, bringing cotyledons above the ground. Epigeal germination. Epicotyl emerges, while cotyledons remain below the soil. Hypogeal germination. Prevents germination even under favorable conditions. Seed dormancy. The production of viable seeds without pollination, resulting in genetically identical offspring. Apomixis. Inhibits germination due to unfavorable conditions. Seed quiescence. Only asexual reproduction. Obligate dormancy. Both sexual and asexual reproduction. Facultative dormancy. Detaching naturally detachable organs from the mother plant. Separation. Caused by essential germination components being unavailable or by environmental factors during seed development. Primary dormancy. Cutting specialized vegetative structures into sections. Division. Imposed by temperature, light, water, chemicals, or gases. Secondary dormancy. Due to unsuitable environmental factors. Ecodormancy. Aerial plantlet formed in leaf or flower axils. Bulbil. Regulated by physiological factors within the seed. Endodormancy. Treatment to alter or remove the seed coat, improving water and air permeability. Scarification. A thickened root containing stored food. Tuberous root. Involves cutting a portion of a stem, root, or leaf from the parent plant and providing favorable conditions for the development of roots and shoots to form a new plant. Cutting propagation. A miniature corm that develops between old and new corms. Canal. Root section is used for propagation. Root cutting. The part of a plant at the surface of the ground from which new shoots are produced. Crown. Portion of stem is used. Stem cutting. A lateral shoot or branch that develops from the base of the main stem, resembling a shortened, thickened stem with a rosette-like appearance. Offset. Leaf section is used. Leaf cutting. A fleshy, enlarged stem section consisting of one to several nodes. Pseudobulb. Cutting with a leaf and bud used for propagation. Leaf bud cutting. A method where adventitious roots are induced on a stem while still attached to the parent plant. Layering. A portion of a stem is bent and buried in the soil to encourage root formation. Simple layering. A leafy shoot originating from axillary buds at the base of a plant or peduncle of the fruit. Slip. Roots are induced on a stem section above the ground before it is detached and planted. Air layering. A modified stem that grows horizontally along the ground. Stolon. Multiple points along the stem are induced to root, creating a serpentine appearance. Compound or serpentine layering. Soil is mounded around the base of a plant to induce rooting along the stem. Mound or stool layering. A trench is dug, and a portion of the stem is buried, encouraging root growth. Trench layering. Method of joining parts of plants to promote their union and growth as one plant. Grafting. Used in species with a thick bark that can be easily separated from the wood. Patch budding. Detached shoot with dormant buds becomes the upper portion of the graft. Scion. Lower portion that develops into the root system. Rootstock. Performed on a stem with actively developing bark that can be separated from the wood. Shield or T-budding. A stem piece between scion and rootstock to avoid incompatibility or control growth. Interstock. Used when stem bark adheres closely to its wood, often due to natural characteristics or growing conditions. Chip budding. A technique where plants are united while growing on their roots, enabling successful grafting even when plants are not in close proximity. Enarching. Changing the top of established plants to more desirable ones. Topworking. Detached scion with buds is grafted onto the stalk. Splice grafting. Scion is grafted onto the stalk by splitting its top. Cleft grafting. Basal portion of scion is joined at the side of the stalk. Side grafting. Scion is grafted onto a stalk with larger stem diameter. Bark grafting. Genetic makeup of a plant that determines its potential yield and other characteristics. Genotype. Similar to grafting, involving joining two plant parts, usually with one bud, and a small section of bark. Budding. External factors influencing plant growth, such as climate, soil,